The human genome, or all the DNA in an individual, is actually very, very similar between every single human on the planet. We have 99.9% of our DNA is exactly the same. That means the ATCG sequence is the same. And in every cell of each individual's body, we have the same set of genes, which is really interesting, right? That you have the same set of genes, the same DNA in your eye, that you have in your liver, that you have in your skin, that you have in, your heart. So it's the same DNA all over. And so we'll look at in this unit why different cells are different ways if it has all the same DNA. The only cells that have different DNA are your reproductive cells, not the ovaries and testes themselves, but the eggs and sperm. And those only have half of the amount of genes that the other body cells have. So the question is, why aren't we all exactly the same? And that's because of the few variations we do have, which are called alleles. So alleles are different versions of the genes. So it's that 0.1% difference between individuals in the human population. Alleles result from mistakes in DNA copying during S phase of DNA replication. Those are called mutations, the mistake. Now, mutations are not good or bad. They can be neutral or they can have an effect. The second thing is not just differences in the genes themselves, but difference in the regulation of genes. So we're looking at, is the gene on or off? On means that it is expressed, meaning that the gene is being read to make some product that does something. How much is the gene turned on or off? Where is the gene on or off? And that's one of those things that makes our cells different in different body parts. And then also your environment and experiences can also influence this. So the environment can increase or decrease gene expression, even without changing the genes themselves. How do you get your actual traits, like the characteristics of your body and the cell's function and all that? So it's the combination of which alleles, which versions of genes you have, and then also how those genes are expressed. This gives you the physical and behavioral traits of yourself. This also goes for all organisms, not just humans. Whether they're single-celled bacteria, they're seaweed in the ocean, they're a mushroom, or there's some kind of other animal like a cat. In this unit, we're going to look at genetic diseases, specifically sickle cell anemia. Just to note, most diseases are influenced by inherited genetics, meaning the gene combos you got from your parents, but most diseases are not genetic diseases. Most of those are resulting from many genes all acting together. There's environmental influence going on, which might alter gene regulation, and there's also probability involved. So just because you have higher probability, like you have genetics for higher probability of Alzheimer's, does not mean you'll necessarily get it. That's the environment and random chance to some degree. When we talk about genetic diseases, we're looking at specific alleles or versions of a gene that cause that disease. Many of those diseases are heritable, meaning they're passed from parent to offspring. Some genetic diseases are what we'd call spontaneous, which means that they are random changes, and this is during reproduction. So whether it's during the egg or sperm formation or very early stages of actual embryonic development. So that genetic change, that genetic disease would be specific to that individual that's born, but not passed on from the parents. If we look at what a gene is, you can see in this picture, a gene is just part of the chromosome. So remember, chromosomes are made of DNA and some proteins. Part of that DNA or parts are called genes. And why do we call them that? They have information in them for making a functional product, something that does something in the cell. And a lot of times that product is protein, and sometimes that product is RNA that does the job in the cell. So that's a brief overview of genomes and genes and how they influence genetic disease.